Okay, so in this review video, we're going to be talking about DNA transcription and translation. So to begin, we'll start talking about the central dogma and the basic structure of a DNA. So the central dogma states that information or genetic information travels from DNA to RNA to protein. And the process of DNA to RNA, so going from DNA to RNA, is known as transcription. And the process of going from RNA to protein is known as translation. So in eukaryotes versus prokaryotes, the places in which these occur is different. So in prokaryotes, transcription and translation both occur in the cytoplasm, whereas for eukaryotes, transcription occurs in the nucleus and then translation will occur in the cytoplasm. Okay, so DNA is composed of subunits called nucleotides. And these nucleotides are composed of a deoxyribose sugar, a phosphate group, and a nitrogenous base. So for DNA, we have a hydroxyl group on the 3' carbon, but only a hydrogen on a 2' carbon. But for RNA, we would have an OH, a hydroxyl group on the 2' carbon as well. And that would be the structure for RNA. So for our bases, our nitrogenous bases, we have two different types. One is known as purines, which are going to be adenine and guanosine, and those are double ringed. So if you look at this here, adenine and guanine are double ringed, and then our pyrimidines are going to be thymine and cytosine, which are single ring. So A and T will bond together, and G and C will bond together. For RNA, it's important to note that thymine is replaced by uracil, which is denoted by just a U. If we look at guanine and cytosine bond versus adenine and thymine, we see that guanine and cytosine actually has 3 hydrogen bonds, whereas adenine and thymine only has 2. This means that this bond is stronger and it's harder to break. Another important thing to know about base pairs is the percent of adenine will be equal to the percent of thymine in the DNA, as well as the percent of guanine will be equal to percent of cytosine. So if you know how much adenine there is, there has to be the same amount of thymine because they're complementary. They will always bind to each other. If you think of these bases as kind of like the alphabet, we have in the English alphabet, we have letters going from A through Z, which they'll ultimately make a word, right? And that word will combine with other words to kind of use for communication. And similarly, in genetics, these bases are kind of like the alphabet that are used to make words known as codons. And those words, they relate to a specific amino acid, which then can make a polypeptide chain known as a protein. Okay. So this is kind of how our DNA looks like. We have these bonds right here in blue, known as phosphodiester bonds, and so they're bonding carbon to oxygen and phosphate to oxygen to carbon. Another thing to note here is that DNA is polar. And what I mean by polar is that it's structurally polar. So the one end of the DNA will have 5 prime, and the other end of the DNA will be 3 prime. So for the other strand of the DNA, it's going to be complementary or anti-parallel to it. So if we start off at a 3 prime down here, it's going to be a 5 prime for the other strand, moving up to a 3 prime. So in transcription, what happens is our DNA is used as a template to make complementary RNA. If you look at this diagram here, we have our DNA, and in the red is our RNA. So the first step kind of transcription is known as initiation. And in initiation, what we have is a promoter sequence. And in eukaryotes, that is usually known as a Tata box. And it's just, it's like a base pair sequence that goes 5 prime, TA, TA, A, 3 prime. And that's kind of where our transcription kind of begins, and this is usually with the help of an enzyme known as RNA polymerase. Okay, so once we've initiated our transcription, we have to elongate our RNA strands. So right here, we'll take a closer look at that. So in the red, we have our DNA, and then here in the blue is our RNA. And notice that for RNA, we are always adding nucleotides to the 3' end. So our enzyme that helps us with this kind of is like, it's kind of engulfed in this like that. And so I'll label it RNA polymerase. And this enzyme, what it does is it just kind of goes along the DNA strand. And the strand that is transcribed is known as our template strand. And the strand that is not transcribed is known as our non-template strand. And so our RNA polymerase molecule will be going to the right, and then we'll be adding nucleotides to the 3' end of the RNA. So our RNA transcript, if you see right here, will have complementary bases to that of the DNA template strand. But since we have another strand of DNA right here, those nucleotides that are transcribed will be the same as a non-template strand. Because if you were to take away this RNA and just combine those two DNA strands, the non-template strand would have the complementary bases for the template strand, just like the RNA will. But the only exception here for the RNA is that it has uracil, right? So it has uracil instead of thymines in its bases. So one more thing about this is that when you add nucleotides to a 3' end of the RNA molecule, when those bonds form together, it releases high energy phosphates, which then are used to drive the next, to add the next base. And so one thing that I forgot to mention was what happens to this RNA, like where does this end? So just like how we started this with a promoter sequence, this RNA transcription of the template strand is going to end with a terminator sequence. So going back to this picture here, right? So we started at a promoter, and that had like the Tata box sequence, and eventually once we hit another sequence of bases, it's gonna stop the RNA transcription, and the RNA template is now gonna come off the DNA strand, and we'll go into the next step known as RNA processing. So once we have our RNA transcript, we're going to move on to a process known as RNA processing, and this is only in eukaryotes. In prokaryotes, they don't do this step. So in RNA processing, we have our RNA strand, and we have regions known as exons and introns. Our exons are going to be protein coding. So they're going to have those protein coding bases, and our introns are not. So they're going to be essentially spliced out. So it's known as splicing, and this is done by an enzyme known as spliceosome, I believe. Okay, and that gives us our mature mRNA, and mRNA means messenger RNA. And this is the RNA that is used to make the protein. But before it can do that, we need to add a 5' cap and a poly A tail. So the 5' cap is a 7-methylguanosine, and it is essential for translation because the ribosome that translates the mRNA, it recognizes it by its 5' cap. And for the poly A tail, it is just a string of 250, sort of rough estimate, of adenine nucleotides that help to transport this mRNA out of the nucleus into the cytoplasm, ultimately where translation occurs. And also both the 5' and the 5' cap and the poly A tail, they help to stabilize the mRNA molecule. So this is kind of one fate of RNA when it's transcribed. However, not all RNA that is transcribed will become a messenger RNA. Some will become tRNA. Actually, most will become tRNA and rRNA. So tRNA is transfer RNA, and rRNA is ribosomal RNA. And these are very important because they are used to translate the mRNA. Okay, so now that we have our mature mRNA for eukaryotes and just RNA for prokaryotes, we move on to a step known as translation, and where we will synthesize a protein out of our mRNA molecule. So how this happens is that we have an mRNA molecule that's shown in the blue here. 5' on this side and 3' here. Our mRNA molecule will be read by what is known as an anticodon. And well, actually, before I talk about that, the way that it's read is that it's read in pairs of 3. So we have AUG, that's known as a codon, or all of these are known as codon. And that codon will specify a specific amino acid. And that amino acid will ultimately be combined with other amino acids to make a protein. And the way it does this is through the help of a tRNA molecule, shown in yellow here, which has a anticodon. Which is complementary bases to our mRNA molecule, shown in blue here. And our anticodon will always be antiparallel to our mRNA molecule. So if we start off here at AUG, since this is going 5 to 3 to mRNA, our tRNA molecule, or our anticodon, will be 3' to 5' going that way. So we have that, right? And then up here, once we have our anticodon pair with our codon, we will get a amino acid that's shown in green here. How we get that amino acid is from this codon chart. So each codon will code for a specific amino acid. And in the case of translation, we will always start at the codon sequence of AUG. That's where our translation will start. And that codes for methionine. Okay. This is all done by the help of a molecule known as a ribosome, which is shown in the gray here. It has two subunits, known as the large subunit and the small subunit. So when this process first begins, our small subunit will first come and recognize our start codon, and then our large subunit will come to form that continuously growing polypeptide chain. And translation, just like transcription, can be divided into 3 steps. So we have our initiation, not promotion. And then we have elongation. And then finally termination. And initiation will start at an AUG codon sequence, as I mentioned before, that codes for methionine. Once that happens, we will start to elongate our polypeptide chain, basically by with more tRNA molecules coming in and it building that polypeptide chain. And finally, we have termination. And the way that translation stops is through, just like how initiation started through a codon sequence of AUG, termination will stop through what is known as a stop codon. And it's just another it's just another codon, but it will specifically specify that ribosome to stop the translation. And if we look at this chart here, we have 3 of those, which is shown. So if you encounter UAA, UAG, or UGA, that means our translation will stop. If we think of like an analogy for translation, and ultimately what it does is synthesize a protein through a series of amino acid sequences. You can think of this like, let's say we have a word protein. Sorry, that's not how you spell protein. We have the word protein. It's made out of an arrangement of the letters P-R-O-T-E-I-N, right? However, we can rearrange this and come up with a completely different word. So we have pointer now. I use the same exact letters, all of them, but I came up with a word that says something completely different than protein. So what I'm trying to say here is that in translation, the sequence of amino acids will specify the specific protein that will form, and basically how that protein will fold and whatnot. So when we're going through translation, it is very important that we have the correct sequence of amino acids, or else it will completely code for something different.